So thank you for the opportunity to give this talk, and apologies that I'm not able to attend the Q&A session. So I'm going to talk about quantitative approaches to support antimicrobial treatment development for neonatal sepsis. And drug development for children is in real crisis despite legislation changes a few years ago. At the time of marketing, only 14% of drugs have a paediatric indication, and of those that don't have a paediatric indication when they come to market, only a quarter ever do in the lifetime of the patent within 12 years. The situation in neonates is even worse. Only 4% of drugs have got a neonatal license when they come to market. So I'm going to start today's talk by talking about a neonatal pivotal trial in late onset sepsis. This was a randomized controlled trial, multi-center, multinational, and it was powered to detect the superiority of meropenem versus standard of care in settings, NICUs where there were high ESBLs. The grant was submitted in 2009, and also there was a meningitis sub-study, NeoMero-2. Now, as you can see, it took us 11 years from submitting the grant to actually publishing the main paper, and even then the full sample size hadn't been reached. We did collect PK/PD as a secondary endpoint, so we collected trough concentrations in all the meropenem arm patients, and at specialist sites we collected 3 optimally timed PK samples, and in the end all the patients in the meropenem arm had at least 1 PK sample, and this was published, as you can see, 2 years before the main trial, incidentally in a higher impact journal. Make of that what you will. So we've recruited all these patients, but only 24 of them had a gram-negative organism for which we could get an MIC, for which we could compare the PK/PD target attainment. And as you can see, it was the wrong way round. The patients who failed the test of cure visit, and these black dots are the 2 babies that sadly died, had better PK/PD target attainment in terms of C-min over MIC or AUC over MIC versus the ones who didn't fail treatment. And of course, this is because there is a limit to what PK target attainment for the antimicrobials can do. And when you're in multi-organ failure and you can't clear the drug, of course your levels are high, but that doesn't stop you from having a bad outcome. The overall result with the lower sample size with the meropenem was not superior to standard of care. So in many ways you could think that this trial was something of a failure. But pivotal trials are not only difficult in neonates, of course we know they're very difficult in adults. Achaogen went bankrupt when one of their ambitious phase 3 trials failed. And clinical PK/PD is confounded by patient status, organism susceptibility and toxicity. In a clinical study we have no control of these things, we just have to hope. This example from NeoMero is not the only one. It's been seen a few times where the PK/PD relationship is in the wrong direction. So what we can learn from this case study and from the literature is that we're not going to be able to do in many cases these big pivotal trials, certainly for new agents. What we need is PK, safety, and then some sort of extrapolation of the PD so that we can do a better job of getting things licensed in babies. So the rest of the talk I'm going to focus on PK, then safety, then PD, and then conclude, focusing on the quantitative approaches. So for PK we need to know how many samples to take and how many patients to recruit and when to take those samples, how to scale for size and age, and how do we account for gestational age and postnatal age, and what age groups does that imply that we should recruit. So we certainly shouldn't be doing opportunistic sampling. It's not optimal, and there's 2 examples here that shows why. Population PK of piperacillin using scavenge samples. We've got weighted residuals here that should be between +2 and -2 if they follow the standard normal distribution, showing that the model was massively variable, and the authors conclude that the scavenge sampling didn't allow them to make a reliable model for dose predictions. A similar result was found in the TIN study for neonatal ciprofloxacin, where a comparison was made in a clinical trial of timed samples and scavenge samples. And what you can see here is that the model based on the scavenge samples and the opportunistic samples gave a much different Cmax than the time samples. So actually, a small number of time samples is better than just throwing lots of noisy data at the problem. So in general, we ought to avoid opportunistic sampling and try to take few samples, but in a well-designed way. So there's two ways to do that. The first way we did, like in Neomero, was in our PK specialist sites. We optimally designed 3 sampling times, and that's, you can see the cloud here. We had a longer dose interval in the very preterm neonates. That's why some patients had their trough at 12 hours. But overall, we captured the dose interval with 3 optimally timed samples. In situations where optimal design is a bit harder and it's not so clear what the pre-existing model is, you can do something, an idea that we got from Catherine Maitland and Alison Thompson, who did a gentamicin study many years ago, to randomize the sampling times across the population. So we've done this a few times, but I give you the example of the Neophospho trial, where we took 2 samples, but at different time points, randomized for different patients after an IV dose, and then crossed over to oral. I believe this was one of the first ever crossover studies to be done in neonates. And then, as you can see, we covered the whole dose interval despite only taking 2 samples from each child from each dose interval. So in general, 3 to 4 samples per child is sufficient, and you can confirm if those samples are at good sampling times with simulation estimation. There are some pitfalls when it comes to fitting the model and deciding which model to simulate from, and I've just taken some examples from the literature in 2022. So all these 4 examples were all published this year. The first pitfall is if you make up your own mathematical modelling functions to scale clearance and volume distribution things, you may make a mistake. And there were 2 consecutive articles in JAC earlier this year that did just that. So this was a study on Flumoxef, where if you extrapolate out from their reported parameters, the adult clearance was reached by 4 weeks of age. Of course, it turned out, after we wrote to the authors, that this was a typo, and the B and the A had been mixed up. Similarly, the creatinine function in this paper showed that if the creatinine went up to just 100 from a median of 73, the clearance would be 12 litres per hour. Again, an adult clearance with a small change of creatinine, completely physiologically implausible. So again, this turned out to be a typographical error in the paper. Another couple of examples is this paper on vancomycin, where there was no weight scaling on the peripheral compartments. And so what that meant is that the terminal half-life, if we included scaling, was 7 hours rather than 15 hours with the reported model. So if we start to extrapolate away from the central patient, we get very different half-lives if we don't biologically inform our scaling. Here's another paper where the median age range of the patients was 1 to 3 days, and if we extrapolated from this model, which has postnatal age as a function in the clearance model, the adult clearance would be reached by 1 year of age. So this model would be very dangerous to try and extrapolate outside the studied age range. Now, recruitment to novel agents is really challenging. So here's clinicaltrials.gov, and you can see terminated. So we're never going to be able to cover every possible combination of postnatal age and postmenstrual age. And that's a couple of those terminated trials. That's what they were trying to do. You can see there's lots of cohorts with different gestation and different postnatal ages. And maybe this was one of the contributing factors to the studies not being able to fill up all of these pre-specified age groups. So what we've found is that if we use biological prior information on how we think clearance should scale, so this is what we could call PK extrapolation, we can fix the weight and the post-menstrual age model using these biological priors, and then estimate a creatinine, if it's a renally cleared drug, and a postnatal age effect. And we showed this is a very rare study, the NeoGent study, published in 2016, but in this study we had externally collected data. So we prospectively ran a multi-center trial and collected almost 200 babies worth of PK data. We then used our model that was based on other data, historical data, with these biological priors. We weren't chasing a good fit to the model, we were chasing a model which was biologically informed, and the predictive performance of this model on this external data set was much better than all of the other published models, despite the fact some of them were from much bigger data sets. The main thing that's come out of our neonatal research over recent years is this importance of the first week of life. So after birth, clearance radically increases over the first 7 to 10 days. And this is the maturation of clearance we saw in that neophosphor trial. The beauty of it was, of course, we longitudinally sampled patients after their first dose and after their oral step down. So now I'd like to move on to safety, and I've only got one slide on safety. In general, you need to study 300 patients to see an event, to have a 95% chance of seeing an event occurring in 1 in 100 people. So this is a well-known statistical fact based on the Poisson distribution, but in general, it's going to be hard for us to recruit that many patients. We must obviously do causality assessment and look at all adverse events in PK, but there is an opportunity for randomization. So again, in neophosphor, we did have a potentially common safety event of high sodium load from the fosfomycin formulation. So we randomized patients to receive standard of care or standard of care plus fosfomycin, and we're able to show that this fosfomycin overload was safe. Safety is really going to have to be looked at in post-marketing surveillance, however, if we need so many hundreds of patients before licensing. And I think there's a real opportunity here in electronic health records. And so, for example, at our hospital at Great Ormond Street, we've got this data lake and an ethical approval to capture anonymized data, which we can then analyze once these drugs get into use. So I'd now like to go on to pharmacodynamics or efficacy, and in this I'd like to define how we set the PK/PD index, and then how we derive it, then how do we set the dose with PK and the PK/PD index, and a little bit on how we can potentially replace animal models that are used to set the PK/PD index. So as you're probably aware, the PK/PD index relates the pharmacokinetic curve with the bacterial susceptibility measured by the minimum inhibitory concentration, the MIC, and we want to know two questions. We've got a qualitative question: which index to choose? Is it Cmax over MIC, AUC over MIC, or time above MIC? And then we've got a quantitative value thing to measure is, once we know the index that we're aiming for, what value of that index do we need? What AUC to MIC ratio do we need for efficacy? Now, this can be done from clinical studies, and there's a classic example from oral candida and fluconazole AUC-MIC, which is a very clean example where it was shown that AUC-MIC was the marker, and the probability of cure nicely tracks that ratio of the drug concentrations to the MIC. And there are a range of published examples where an animal or an in vitro, usually an animal actually, model of PK/PD index scales and is similar to the clinically derived target. But as I said at the start, clinically derived targets do need to be taken with a pinch of salt. We need to be careful of potential for publication bias, disease state, and the real difficulty in defining these clinically derived targets. But what it means is that in general, PK/PD targets are set in animals using dose fractionation studies. So how did they set the PK/PD index of cafidrocol? Well, they used lots of mice, and this was mainly the neutropenic mouse thigh model, with lots of different treatment frequencies and doses and organisms, and then they plotted the AUC-MIC, Cmax-MIC, percent time above MIC against the change in colony forming units per thigh for cefepime and cafidrocol, and then they use the R-squared value, a measure of fit, to pick a PK/PD index. And so they found that the fraction time above MIC was chosen as the index, and then the quantitative measure depended on the organism, and so conservatively you'd want to aim for high 80s% time above MIC for cefadrochol to cover most of the organisms that you're interested in. So this is how UCAS goes about setting doses. So they take preclinical PK/PD, clinical if it's possible, look at distributions of MICs, and then take published PK models to set a PK/PD breakpoint. So it accounts for clinical and preclinical information, but in general it's the preclinical that does the heavy lifting for most drugs. Now, in our lab we use the hollow fibre infection model, and one of the things that we think is that we could probably replace these dose fractionation studies where the mouse is rendered neutropenic and perhaps the immune component is very small. Could we do similar dose escalation studies in vitro in the hollow fibre system and derive a PK/PD index? And this is our result for fosfomycin, which shows not quite the same, but possibly a scaling where we'll be able to extrapolate between the hollow fibre and the mouse and reduce the need for doing lots of animal experiments. Now, of course, hollow fibre isn't just limited to these dose fractionation studies, and another example of where we've used it is in the study of combinations. So FICI is a measure of additivity or synergy of two agents together, or even three agents, because it can all be added up. If the FICI is less than 0.5, this indicates synergy, and we tested against E. coli and Klebsiella isolates that were resistant to standard first-line agents, so ampicillin, gentamicin, cefotaxime. These organisms were all resistant to these agents on their own, but when we combine them, and especially when we look at triple combinations and add in the beta-lactamase inhibitor sulbactam, we start to get synergy even in a Klebsiella, one of which was a carbapenem-resistant organism as well. We can then use the hollow fibre to really challenge these antibiotic combinations against these organisms. So what we've done here is we've actually allowed the organisms to grow up to a stationary phase before starting therapy. And what you can see is that this combination of cefotaxime, sulbactam plus gentamicin in the standard 3-to-1 combination that's commercially available in countries like India can completely eliminate the hollow fibre cartridge, at least within 7 days, using standard neonatal dosing. And it even worked on the carbapenem-resistant Klebsiella strain. So to conclude, extrapolation in PK means that we need to use biologically informed prior information to design and our sampling and set the sample size. We can do standardize the parameterization, decreases the risk of errors and increases the interpretability of model parameters and increases the extrapolation potential. And none of this was really our ideas. We've just followed the method proposed by Catherine Sherwin's group. In terms of safety, well, our extrapolation is that we can't treat neonates in isolation. We're never going to get big enough data sets to fully confirm safety before licensing. So pre-licensing trials are likely going to be too small with neonates alone. So we're going to rely on juvenile toxicity data, etc. Where possible, you can randomize to rule out common expected adverse effects, but I think there's a real role of real-world evidence and phase 4 post-marketing surveillance that electronic healthcare records could contribute to. For PD, extrapolation is absolutely necessary in most cases, as you've seen from the difficulty and the failures of running pivotal trials. We must try and replace animal models at least for dose fractionation, and we can do that with the hollow fibre. And in my view, the hollow fibre infection model ought to be more broadly utilised. So I'd like to thank you for your attention and thank my students and postdocs who've done most of the work presented here and our funders as well. Thank you.